Hello, everyone. Welcome to another episode of Research Digest podcast. In this episode, we'll talk about RNA biology and a recently developed cool method to profile RNA dynamics to study many different aspects of the biology. So, let me introduce you with our guest today. I'm happy to have Dr. Nadiya Khyzha with me, postdoctoral fellow at Fred Hutchinson Cancer Center based in Seattle, United States. Nadiya, welcome to this podcast.
Thank you for having me.
It's my pleasure. So, first, could you please share briefly your research journey with us, your PhD and postdoc, everything, and briefly what you worked on those steps?
Yeah, for sure. So the main questions that I've been really interested in, I'd say starting from undergrad and going through my PhD and also my postdoc, is understanding how the genome is regulated. As you may know, we all have kind of like a static genetic code, but that isn't really determining how that genome is expressed. And there's a lot of different regulatory layers to this. One of them is the chromatin environment and also some of the RNA aspects of it as well. And so I got really interested in this idea of genetic regulation or gene regulation or epigenetics while I was an undergrad. And we had this one slide or one little area when I was during undergrad, and I was talking about this concept of long non-coding RNAs. And it was maybe at that time still a not well understood topic, but I remember one of the instructors was talking about this long non-coding RNA called EXIST. And I thought it was the coolest thing in the world because essentially it's this long non-coding RNA that is, it doesn't produce a protein and it actually just sticks around on chromatin, and it is expressed on one of the X chromosomes in females. And what it does is it has the capacity to shut down that whole chromosome. And so I thought wow, these long non-coding RNAs are really powerful and have a lot of potential gene regulatory functions.
And that's how the name came, right? X chromosome inactivation.
Exactly.
Yeah. That's wonderful. Yeah. I also love that.
Yeah, so that was a really big motivation for my PhD work. So I was at the University of Toronto, and there I was working with Dr. Jason Fish, and he's a vascular biology specialist, but I was working on looking at long non-coding RNAs and seeing how they might play a role in modulating vascular inflammation. So the main problem is that our vasculature, we have these cells that line the vasculature, and these cells are kind of able to sense the inflammatory environment, and they will respond to it. And in a short, acute sense, this kind of a response is good because it will help you detect whether you have a pathogen in your bloodstream and mount an immune response. But if prolonged, this inflammatory response is actually very dangerous. It can lead to chronic diseases. And so there really needs to be a good way to modulate this vascular inflammation. And so I was trying to see what are the potential long non-coding RNAs that are regulated by this inflammatory process and see whether some of them could be responsible for tuning down this vascular inflammation.
Right. Okay. Let me pause you there for a moment. I want to pull up that wonderful paper that you published out of that study with long non-coding RNA. It was published in 2019. So also briefly tell us what you did in that paper.
Yeah. So a big part of this was trying to figure out what long non-coding RNAs might be functional during vascular inflammation. So the easy part in the long non-coding RNA field is doing a microarray or an RNA-seq and identifying a bunch of long non-coding RNAs that change in expression. or those levels change. But the much harder question is figuring out which ones are functional and what they're doing.
Right.
So a big part of it was there's been some literature in the field suggesting that long non-coding RNAs act on their neighboring genes and they can regulate whatever's nearby. So I designed this computational approach to essentially define mRNA, long non-coding RNA pairs that are kind of within close proximity to each other. And then I found one long non-coding RNA that was pretty close to a major chemokine cluster, including CCL2. So these chemokines are important for recruiting monocytes to the vasculature.
Yeah.
And I found that this long non-coding RNA actually regulated the levels of the cytokine that was near its CCL2. And so I thought that was a pretty exciting observation. But a big part of what I learned while I was working on long non-coding RNA biology was just how hard it is to study these long non-coding RNAs and just how important technology development is. So these long non-coding RNAs, some of their caveats is that they're not particularly abundant, and so they're very difficult to capture. And methods to study them were kind of emerging as I was still in grad school. And a lot of the times I would open up a paper and look at one of these new methods and then just think to myself, like, ooh, this looks very complicated. I need all of these special reagents. I need to use like 20 million cells per condition. And I don't know if it's going to work. Do I really want to invest months potentially to get this assay to work? So that was a kind of something that really stayed with me after I finished my PhD. And that was a big part of why I then moved on during my postdoc to Stephen Henikoff's lab here in Seattle, was because he was quite a pioneer in terms of technology development. And so I thought, I had a pretty good motivation to do some tech dev based on my own personal experiences.
So before you move on to your postdoc part, so I have a question about long non-coding RNA. As a RNA structural biologist, I'm curious, so in your experience, how much of the long non-coding RNA's functions do you think are dependent on the structure and how much on the other, like sequence and other things?
Yeah, I think that will probably depend on what RNA it is interacting with. What we do know for that really canonical kind of example of exist, it definitely has structure to it, and there are different parts of that long non-coding RNA and different structures within it that are known to interact with different proteins. So there's definitely a structural component to it. There's also some long non-coding.
So you're saying it's mainly dependent on the binding partner or the functional partner. Okay, got it. And also, as you are saying, studying these long non-coding RNA LNC RNAs are difficult because they are not translating into proteins. And sometimes based on the translation, a lot of methods depend on the translation, right? They see co-translating proteins and RNAs co-localized. So that means something is happening. We can see inside. But that's not happening for LNC RNAs.
Yeah, long non-coding RNAs are really a black box. So, you'll be able to identify it and you know what its sequence is, but it's really hard to predict what is its function. Exactly. That really is kind of the big challenge in the field. And so usually the kind of things that people look at is trying to figure out what proteins bind to this long non-coding RNA or where is it binding in the genome.
Okay, right. Okay, so after that you joined Henikoff and Ahmad lab. And what are you working on that? What's your research focus there?
Yeah, so I've been working on a lot of technology development,
Mm.
namely in the sphere of next generation sequencing and Illumina based technologies. And I am a little bit of an odd one in the lab where everybody's working on chromatin, but I'm working on the RNA side of things. So the main thing that I have done was develop this method called reverse transcribe entagment,
Right.
or RT&Tag, which
Mhm.
is really a tool that can have very wide applications
Right,
in
right.
terms of what it could do. I kind of had initial ideas of where to take it, but once you develop something or once a project gets going, things just evolve and you start having
Yeah,
applications that you never really intended to in the first
exactly.
place.
It's difficult to predict at the beginning, right? So let me show everyone your wonderful paper that you published in Nature Methods, and its title is Profiling RNA at Chromatin Targets in situ by Antibody Targeted Fragmentation. Um, so, uh, in this paper, you mentioned that in the background section, mapping chromatin-associated RNA remains a challenge, right? So yeah, so we'll listen about this particular method, how it works. But before that, can you tell us why that's still a challenge for us?
Yeah, so if you think about chromatin
Mm.
and what it is comprised of, is it's really a site of transcription.
Mm.
And there is going to be a certain proportion of the chromatin that is just going to be recently transcribed mRNAs that are just a natural kind of byproduct. They are not necessarily functional or not necessarily doing anything.
Right.
And there might be another pool of RNAs that are something like a long non-coding RNA like exist, where it's actually sticking around on chromatin and having some sort of a functional role. So the challenge is that the amount of these nascent transcripts is so high that it almost becomes like finding a needle in a haystack in terms of finding transcripts that are potentially regulatory or functional versus just a byproduct of transcription. And so there have been two main types of methods that others have been using to characterize chromatin-associated RNAs. One of them are these RNA-DNA contact maps. So what they would do is cut RNA and DNA to pieces and then ligate those RNA, DNA pieces together. And whatever is right beside each other is going to get ligated together. And then you can sequence it and essentially be able to tell what piece of RNA is interacting with what piece of DNA. Now, most of what you're going to capture are these mRNAs or these recently transcribed transcripts. So you do have to then try to get ways to reduce the amount of these mRNA signal, and also you can do things like sequence deeper and whatnot. So that's the challenge for that particular type of method. The other type of methods that have been used are immunoprecipitation-based methods or pull-down methods.
Yeah, that's the
And
most commonly used, most probably.
yeah, and with those kind of methods, you're using either probes to target an RNA or some sort of an antibody to a target on chromatin. So it could be like a histone mark or some sort of other protein that's sitting on chromatin. And you lyse the cell or the nuclei open, and then you use antibodies to try to pull out these RNA protein complexes. Now, the challenge with those kind of methods is, although you can do polyA selection to, get rid of the nascent RNA signal, these methods are not that efficient due to just the nature of these kind of pull-downs. And so a lot of the time you're using millions of cells per pull-down condition, and also your signal-to-noise ratio is not very high. And so these kind of techniques had challenges just due to their efficiency.
Okay. So in simple terms, explain a little bit how this reverse transcribe and tagment method, or in brief, shortly, RT and tag method works.
Yeah. So for something like RT and tag, it is a very different style of experiments relative to kind of conventional pull down methods. And it is part of something that's called an antibody mediated enzyme tethering method. And so the way it works is that you are not lysing the nuclei or the cell open, you're just permeabilizing it and adding little holes to it. And instead of trying to pull things out, you're actually adding reagents in.
Okay.
And so you will use antibodies to bind to whatever your target of interest is in the nucleus. And then there's this enzyme that you're using that has a PAG moiety. And PAG will bind to antibodies, and it will essentially tether your enzyme to an antibody. And then you can use that enzyme to do whatever reactions you want to do. So it's a much more efficient way of doing things because you're not losing so much material, and these kind of methods tend to have much nicer signal to noise.
I guess it is more specific as well, right?
Yeah. And you're capturing interactions in a much more of a endogenous environment. So you're potentially avoiding artifacts of interactions that can happen within a lysate. So in the case of RT and tag, it combines two reaction components. One is reverse transcription, and the other one is tagmentation. So through these tethering kind of approaches, one of the things that I'm tethering is an oligo-DT. So that's going to allow for a targeted reverse transcription reaction. And then the other part is tethering this TN5 enzyme. And what's cool about TN5 is that it can add in adapters, or it can just kind of stick them into a piece of RNA, cDNA hybrid, or genomic DNA. But in my case, I'm focusing on the RNA cDNA hybrid. And so once you reverse transcribe your RNA, you're going to get these sequences that are stuck or inserted in. And then you can employ these sequences as PCR landing sites, and then you can use PCR to generate sequencing libraries from that and just amplify them up. So everything is happening within a nuclei, essentially. So you're really not like losing a lot of material. And with this kind of approach, I was able to go down from several million cells that you would use in a RNA immunoprecipitation to 100 or 200,000 nuclei per reaction.
That's wonderful. Yeah.
You no longer have to be culturing cells for a whole week just to be able to do one experiment, which makes things much easier.
Yeah, so that's so wonderful. Tell us about some of the potential applications that maybe you haven't showed, but that's possible to do using this method.
Yeah, I think this is an ongoing thing for me currently at the lab, in the sense that, I had certain intentions for what I developed it from. And because of my long non-coding RNA background, I was thinking about chromatin-associated RNAs. And so that was kind of the first part of it. But it can also be used for really anything that you have an antibody for. So in the paper, I've used it for RNA-protein interactions, RNA-chromatin interactions, as well as modifications that are found on RNA. So things like the M6A modification.
Okay.
The thing that's happening now is that the method is published. So I'm constantly getting emails from people asking, can you use it for this application? Can you use it for this or for that? And I'm only one human being. I've only tested it on so many different things, but it's getting pretty creative as to what people are trying to do. So, right now I'm working with somebody to do RT&Tag and fact sorted cells from from mouse, or we might be doing also something in cells isolated from Drosophila and whatnot, where can you imagine trying to do immunoprecipitations and trying to get millions of cells from something like a Drosophila?
Yeah.
So I think that's where the strengths are going to lie.
I think that's the wonderful part of developing methods, because sometimes the original developer may not realize where it can be used, and other peers and collaborators, they find out more innovative ways to apply that method sometimes.
Yeah. And one thing that I never foresaw coming, but it ended up being kind of a strength of a method, and where I ended up taking it to was compartments. So I was playing around with using this one complex in Drosophila called MSL complex, and I was targeting it because it also interacts with a long non-coding RNA to upregulate one of the X chromosomes in male flies. And something that was kind of at first puzzling was I was picking up a lot more interactions than I was expecting. But then I realized that the reason why that was happening was that although the MSL complex binds directly to the ROX RNA, the MSL complex is going to bind all throughout the X chromosome at specific sites, but still it's going to have many binding sites. And what I was actually picking up were some RNAs that were just being transcribed from the regions near where the complex was binding. So it made me realize that this is actually kind of a proximity labeling method, which I didn't think of before. And so as I was kind of stepping back and, how should I say this? It took me like a year or two to get the method to work. And that had its own challenges. But the true challenge came after the method was published. And I was thinking, what do I do with it? Like, what is really the best application of it? Which is always the challenge with method development. And so I was kind of sitting back and trying to think, what is really like a missing piece in the field, and what can I really leverage in this method? And I thought compartments would be a great fit because of this proximity labeling aspect of RT&Tag.
Exactly. Yeah. So more recently, you published another wonderful paper where you advanced this method and developed SLAM-RT&Tag. And here you described this method, which we can use. Which we can use to kind of profile RNA dynamics within nuclear compartments, right? So tell us more about that. What is nuclear compartment, and why this study is important, and how did you apply your particularly SLAM-RT&Tag method to study that?
Yeah, for sure. So compartments is something that happens both in the nucleus and in the cytoplasm. And it really, I think
this is more commonly known. A lot of people know about that, but the nuclear compartments is rare, I guess. And there is no less study done on that part, I think.
So we all actually learned about one nuclear compartment, like back in middle school and high school, which is the nucleolus, actually. Exactly. But I think we always think of it as almost like a little suborganelle. We never think of it as a phase-separated body when we're much younger in our journey. So essentially, the nucleus is not homogeneous. It is highly structured and contains all of these different little compartments. And what these compartments are is essentially regions where you're concentrating specific reaction components. And in the case of the nucleus, a lot of the time, these are implicated in RNA processing. So taking the nucleolus as an example, it's actually a very structured nuclear compartment, and it forms over the genomic region that it codes for ribosomal RNAs. And these are ribosomal RNAs, they're not like a regular mRNA. It requires a very specific set of processing steps. And so this nucleolus actually basically contains many of the different proteins that are necessary for the biogenesis of ribosomal RNAs. And there are other examples like this as well. So the one that the Hennekoff lab really likes is the histone locus body. And what's really cool is that our histone genes are actually different from a regular mRNA. They don't have a poly-A tail. And instead, they have these stem loops. And so you need very specific proteins that are going to be able to process that particular RNA. And these compartments is where it all happens. So some of the other compartments that exist are things like the nuclear speckle, which is something that I got really interested in. And a big part of that is that if you ever look at a stain of the nuclei that is labeled with an oligo-DT probe, so basically capturing all polyadenylated RNA, you actually see a very strong accumulation of RNA within these nuclear speckles.
Okay.
It's very stark. And there's been some work in the past in terms of finding individual RNAs that localize to these nuclear speckles, but by and large, it was still quite a mystery as to what these RNAs are, or why they're there, or what are they doing? And what we knew about nuclear speckles is that they contain splicing factors, as well as some factors that are involved in shuttling RNA out into the cytoplasm. Right. And there have been a couple of different models as to what nuclear speckles do, but I was interested to know what are these RNAs. And a big reason why people couldn't figure this out is that you can't just isolate a nuclear speckle. These compartments, they're not like a true organelle in that they're not membrane bound. If you try to isolate it, it'll kind of just fall apart. And so this is where these kind of proximity labeling based methods are very powerful. And so this is where I used RT&Tag. And what I found was that, yes, these transcripts are polyadenylated, but they also had a ton of intronic signal to them. And so that was kind of puzzling. And essentially what I realized it meant is that these transcripts are incompletely spliced. And so the way that splicing works within the nucleus is that some of it is co-transcriptional and will happen while the RNA is being transcribed, but not all introns are going to be removed that way, and some are going to stick around and will need to be removed post-transcriptionally. And so essentially the transcripts that end up in the nuclear speckles have these features of post-transcriptional splicing. And so I thought that was very interesting, and kind of to really further prove this point, I was doing some other experiments, and one of them was trying to see whether these transcripts are stored in the nuclear speckle or whether they're transient within it. And this came from some kind of earlier observations or this kind of general thought in the field that nuclear speckles are just storage sites. And this is where SLAM-RT&Tag came in, where you can label cells with analogs of nucleotides. So in my case, it's this S4U nucleotides. And then this S4U gets incorporated into RNA. And then you can do some reactions with this S4U to modify it in a way that it confuses the reverse transcriptase and it adds in a wrong nucleotide in your cDNA sequence. So I combined this metabolic labeling kind of trick with my RT&Tag method. And then what I've done was profile half-lives of RNA inside of nuclear speckles and also in the nucleoplasm, and then compare the two. And so the idea is that if a transcript is kind of stuck and is persistent within a compartment, then its half-life is going to be purely explained by its overall stability. But if a transcript is transient within a compartment, then its half-life in the compartment is going to be shorter than its overall half-life to account for that export factor. And so what I found was that actually most transcripts within nuclear speckles are transient. So I thought this was really nice because it went against this really cemented in the field idea of them being storage sites, and really saying that maybe they have more of a functional role in splicing of these transcripts.
Yeah. So they are not just storing, so they are kind of in the middle of a function, and sometimes they might be another station for a long journey, right?
Exactly. Yeah, the big features of these incompletely spliced introns is that they're hard to splice. They have features that make it challenging.
The transcript that are stored there, they might be partially spliced, fully spliced, or in any steps of their post-transcriptional modification, right?
Yeah. So with the RT&Tag method, it does have some biases in terms of the signal that it can capture, which is that most of it is from the 3' and the 3' end of the RNA. And also it can pick up some introns because introns tend to have some stretches of A's inside of them. So this is where, my tech dev journey is never really finished. So I've been working on a lot of follow-up methods, and one of them is trying to get the full length transcript information in the speckle. And most of these transcripts are quite incompletely spliced. Let's just put it that way. Moderately to incompletely spliced.
Wonderful. Yeah, I hope you will be able to achieve what you're targeting with this method. And I found one quotation by your boss, Dr. Hanikov, and I really loved it. He said, science is always changing. You can just sit back and relax and say, I'll do this over and over again. That's what makes it so fun. It was really interesting to me.
Yeah, that is very much, I would say, the motto in the lab, where because we do so much tech dev, the idea is that if there's some sort of a problem in biology that you're interested in, if people couldn't answer it before due to technological limitations, well, there you go. That's the perfect question to address. So that is one way I always approach science, is trying to, not just develop RT&Tag, but to continue developing methods throughout my career.
All right. That's so wonderful. Okay, Nadiya, finally, my last question is, do you have any final take-home message for our audience or any suggestion for career or young scientists, let's say?
Yeah, well, actually, I think that quote that you mentioned was a big part of one of my career advices, which is, you have to kind of be flexible, I think. And I think there's sometimes pressure when you're very young to have also like everything figured out and to know exactly what you want to study and kind of have an idea of what you want your ideal career is or what you want to achieve. But once you actually go into science, once you start working on a project, you kind of realize that there's a lot of interesting things that you never really considered, and projects always take very much like unexpected turns. And so you have to just kind of be flexible and keep moving with the field and keep advancing and just pivot whenever you need to. And I think it's really important because it kind of stops this kind of like analysis paralysis where it's like, oh, I don't have everything in my life figured out, so I don't know what to do. Sometimes you just have to like do things and figure it out as you go. I think one thing you can take away from this whole my career journey is, I started with long non-coding RNAs to tag dev, working in compartments, and although it's all in the kind of the sphere of gene regulation and RNA biology, it's deviated quite a bit and kind of just goes with the flow.
Right.
So I think that's important. And
Wonderful.
I think if there's one more piece of advice, it's
Sure.
have a life outside of the lab as well. I think it's very important to have other things to balance you out when things aren't working in the lab, and to just kind of have something that is moving in your life, because sometimes you do reach points where things are not working for months. So it's always good to have something to balance.
Okay, that's a wonderful message. So like sometimes some research projects are like a foggy road. If you move ahead, if you keep going, then you can figure out what's next, right? So that's kind of resonate to your message. Like you have to be flexible and keep moving, right? With motivation.
Yeah, for sure. Tech dev is pretty high risk, high reward style of doing science. I mean, RT&Tag didn't work for a whole year. Okay. So at that point in time, it was like, I have nothing to publish, right, potentially. But eventually, it just kind of clicked, and then everything moved very quickly. So that's just the nature of technology development style projects.
There will be a lot of challenges, uncertainty, but if we try enough, hopefully, it will work someday. All right. Thank you so much, Nadiya, for coming to this platform and sharing your wonderful research and messages and everything. Thank you so much.
Yeah, thank you for having me.